what is cell culture, different type of cell culture like continuous cell culture, primary cell culture, biohazards, different safety laboratory practices, biohazards, and different biosafety levels. Students, I hope you are enjoying the course and learn a lot about the topic. Now it's the time to introduce our speaker. It is my pleasure to introduce Dr. Sanchita Dotto as our today's speaker. Dr. Dotto has completed her MSc from Kalyan University and received her PhD degree from Jadavpur University. Dr. Dotto has received the prestigious D.S. Kothari Fellowship and worked as postdoctoral research fellow in the University of Calcutta. Presently, she is working as ICMR Research Associate at the Department of Pharmaceutical Technology, Jadavpur University. Dr. Dotto has published her scientific contribution in different reputed journals like Acta Tropica, Future Medicinal Chemistry, Brazilian Journal of Infectious Disease, and like on. PG Department of Zoology warmly welcomes you, Dr. Dotto. Please unmute yourself and proceed with your presentation. We are eager to hear you. Over to you, Dr. Dotto.
Good evening, everyone. Shomgita has already introduced me to all of you. So I hope that we all are biologists here. Right? We all are biologists, and in future, most of us are going to do in research work. Some of us will go for academic line, like school teaching, college teaching, and all those things. But I believe that many of you will go for research work and be a successful researcher. So in biology, it is not only one subject like geography or physics. Many different sub-branches are there in biology, like zoology, physiology, botany, microbiology, biochemistry, and so on. So we all study different topics of biology, but when we go for research, we come and meet together. Because in research, there is no biology, zoology, physiology separate. We all of us almost do same techniques. Maybe the topic can be different, but techniques will be same. And when we go for biological research, the most common and basic technique of biological research, any type of biological research, whether cancer biology, molecular biology, microbiology, neuroscience, any kind of biological research, the basic technique that we almost all of us have to do is cell culture. Cell culture is the base of biological research. And in cell culture, when we go for like cancer study, we have to do cell culture. When we want to test the different developed drugs or any natural extracts or any kind of molecular study like immunological study or genetical study, cloning or anything, we have to take our basic step, cell culture. So for cell culture, what is cell culture? We first have to know that what is cell culture. So I'm sharing my PPT now. So first we have to know, I know that all of you have gone through this as the workshop is going on, you have learned that what is cell culture. So still I will just give a brief introduction to what is cell culture and all those things, and I will go to my main topic, which is the contamination, biological contamination of cell culture. So now, what is cell culture? Cell culture is the process by which cells are grown under controlled condition, generally outside their natural environment. Their natural environment can be plant body, can be animal body. When we take out the tissue or cell from plant system or animal system and grow it in artificial condition in our laboratory, then it is known as cell culture. So cell culture can be both from plant or from animal. But basically, when we make the cell culture characterize the cell culture, we say that cell culture are three different types. One is primary, one is secondary, and cell line. What is primary cell culture? It is directly obtained from the plant or animal tissue. Like if we take out blood from our body and isolate the IBC or the monocytes from the blood, then plate it in culture dish and culture it with artificial culture media and artificial culture condition, then it is known as primary cell culture. And when we take cells from primary cell culture and subculture it in another petri dish and maintain it for some days and do some experiments with it, then it is known as secondary cell culture. And when this cell, normal human cell, will be cloned with cancerous cell so that it can grow generation after generation for an extended period of time. Normal healthy cells cannot grow for longer time. Maybe 2 or 3 times after 2 or 3 subculture, it will start degrading. It will go to destroy. But in cell line, as we clone it with cancerous cell, it will be growing for enormous period of time. And this is what generally we need in our biological study, to grow cells for longer period of time so that we can do different types of experiments with it. So that is why biologists created cell lines, which are normal cells with different types of characteristics fused with cancerous cells, cloned with cancerous cells, which so that it can grow for extended period of times. So here 2 pictures are showing. One is primary cell, which is blood cell here, and this is HeLa cell line. This is a cervical cancer cell line collected from cervical cancer patient, and it is very useful in recent studies, and we use it very frequently for our biological studies. Now, cell culture is a very sensitive technique. Like other experiments in our open laboratory, whether dust is there, whether it is not neat, but still we can do DNA extraction, DNA gel we can run, we can do protein extraction, protein gel we can run, or Western blotting, or many other ELISA or different kind of other biological techniques we can do on our laboratory desk. But cell culture we cannot do on our laboratory desk. We need a completely separate room to do our cell culture, which is known as culture room. No other experiments can be done there, which is exclusively known as culture room. And in that cell culture room, a laminar hood should be used, inside which all the cell culture techniques should be exclusively done inside the laminar hood. And there should be a incubator, which is the home of all these cells. We cannot just keep cells on our desk or inside even laminar hood. We have to keep the cells in incubator. Incubator is what? It is a system, a machine where it is 37 degrees Celsius temperature is maintained and 5% CO2 carbon dioxide level is maintained. And it is a moist environment. We keep a water bath here to keep the machine moist. So this is the home of almost all the cell lines for almost all the cells. This is the home. So we must have laminar hood, incubator, and another very important thing in a cell culture room is microscope. So in microscope also we have to have in cell culture room, so that anytime we are doing culture we can check what is going on, because cells we cannot see with our naked eye. So we have to use the microscope. We have to look under the microscope whether the cells are okay, they are healthy, they are malnutrition, or what is the condition. So these are the equipments of our cell culture room. This is some plastic wares or pipette mans, all those things are inside laminar hood, and beside this the incubator is there. And refrigerator is also important for our cell culture, because our media, cell culture media, and some other drugs or antibiotics that we use for cell culture should always be kept in the refrigerator. Along with this, as I told, the microscope, common light microscope is also very important for a cell culture laboratory. But why all these precautions for cell culture? What is so special about cell culture? Why we just cannot do it outside in our common lab on the workbench? Why we cannot do? Because of contamination. Contamination is a very common word. It is not applied only for cell culture. It can be like food contamination. It can be like water contamination. Our day-to-day life also uh, we face different kind of contamination problem, like bread gets spoiled with fungal contamination. Sometimes we in many places water also drinking water also get contaminated. So, in cell culture, almost all biology's nightmare is contamination. So, what is contamination? In general term, any unwanted or presence of any unwanted substance or impurity is called contamination. And when in culture media in which we are growing our cells, in that media, any kind of impurity or unwanted substance comes, then we call the culture media is contaminated. And it is a very crucial thing. We have to control the contamination, otherwise no experimental procedure can be possible with the contaminated culture. And what is contaminant? Contaminant is the impurity that causes the contamination. It is called contaminants. Contaminants can be both chemical as well as biological. Chemical contaminants like many are there, like any presence of any particular unwanted salt or presence of any ions, metal ions, or any plastic or any kind of toxin. Presence of all these things are chemical contaminant. And biological contaminants are like bacteria, presence of virus, bacteria, yeast, mold, etc. in our cell culture media is biological contaminants. Now, chemical contaminants, the way we set up our biological experiments, the way we filter our media and use everything sterile, normal basic precautions, the way we take in our cell culture system, normal SOP, the standard protocol that we follow in our cell culture system, chemical contaminants we can avoid very easily. These are manageable. But biological contaminants, though we take several different steps to prevent these biological contaminants, but still sometimes unintentional mistakes causes these biological contaminants, and it spoiled our whole experiments. So for biological contaminants, uh, there are mainly 5 types of biological contaminants in our cell culture system, which are bacteria, mold, yeast, virus, and microplasma. Our biological works, we usually one is very lengthy. We usually maybe let in from flask. We usually grow cells inside flask, but when we go for doing some experiment, like we want to check the efficacy of one particular plant extract on the cells, on particular cancer cell, whether that plant extract can prevent the cancerous property of that cell. So what we do, we take cells out from the flask and put it in some petri dish or some plates. Put it in some petri dish or plates. Then we give. Then we give what we give the plant extracts to the. We give the extracts to the cells, and then we keep it again overnight. Then maybe another second dose of treatment we give. So like this, like 48 to 72 hours, maybe we are going to keep the cells in culture for to complete only one experiment. So it is a lengthy procedure, and we are opening the culture and adding some drugs, again keeping it incubator for some time, then again taking it out, again giving some drugs. So like this, there are huge chance of contamination. So if at the end of the day, maybe after 48 hours, if we see that the culture got contaminated, so all our efforts will go in vain. There will be no expected result as we expected, or whatever will be the outcome of the experiment. We will not get the right result from this experiment. So we have to be very cautious about all this procedure, cell culture procedure. When we are doing cell culture procedure, any experiment with the cell lines, we have to be very cautious about the contamination, particularly biological contaminations. Now, what are the causes, different causes of contamination? Though we take different steps as a precautionary measure, but still, for any our unintentional mistakes, all this contamination happen. But how? What are the causes of this contamination? It can be from the culture room air. Though we keep separate rooms, culture room totally separate, but exclusively for cell culture, but still, we human being, when we laboratory workers will go inside and come out, our sneezing, our coughing, or from our talking also, the air inside room can get contaminated. Then, secondly, from our hands, the biologist who is working inside room, from the hands and clothes of the biologist, also the contamination can transfer into the culture. And for cell culture, we use what? We use liquid media exclusively for the cell culture. And for this, this is where the cells can grow. And as a nutrition of the cells, we add fecal cut serum, FCS, which is highly nutritious. This is the nutrition of the cells. So if the media and the FCS is not properly sterilized, how we used to sterilize them? We used to filter it through 0.22 micron filter paper, so that if any contaminant will be there, it will be removed. So if any after doing all this filtration, still if any small contaminant remain in the media or FCS, it can destroy the full culture. And the plastic wares that we use inside the laminar hood for cell culture, like tips, all those tips that we use for subculturing, the tubes where we take the cells, then flasks where we maintain the cells, if these are not properly sterilized, all those things also can cause contamination. So now going to the different types of contaminant. Firstly, we'll talk about bacteria. This is the most common type of contamination. Bacteria, mainly E. coli and Bacillus, these are the two groups of bacteria which causes contaminations. Bacteria, as we know, it is a large group of unicellular microorganisms. There are many different types of bacteria, and the diameter of these bacteria are very small, few micrometer, and it can be different. It can be of different shape. It can be round like E. coli, or spiral or rod shaped. Different kind of bacteria can be there. Every type of bacteria can cause cell culture contamination. Bacterial size is very small compared to the cell lines or human cells or whatever cells we are growing. Bacterial size is much smaller than all those cells. Like only 1 to 10 micrometer, if bacteria size is only 1 to 10 micrometer, then our cell line or cell size will be like 10 times or 100 times more than this, 10 to 800 micrometer. So when first bacterial infection, if any culture get contaminated with bacteria, initially what we see with our naked eye, we cannot see the culture any difference. We will be subculturing, we will be maintaining the culture. will be, but we will not see it initially with our naked eye. But if we regularly put it under microscope, then we can understand when the contamination is starting. If we keep the flask under microscope, then we can see like dots, several dots. Initially, when the infection just started, then the amount will be not much, small, different small dots we will see. And as it is very small, we will sometimes, most of the time, we consider it as maybe cell debris, maybe few cells broke down and the debris are there. That is why it is looking like those dots are appearing. But bacteria, if it is cell debris, really cell debris, then it's okay. The culture will not get infected. The culture will be maintained properly, and maybe after 1 or 2 times subculturing, we will remove the debris. But when it is really bacteria has contaminated the culture, then after 2 or 3 subculturing, we will see the debris are increasing. Under microscope, we will see that debris, all those dots are increasing in number. And after few subculture procedure, we will see what? We will see with our naked eye. How we will see in our naked eye? The whole flask or petri dish where we are maintaining the culture will turn turbid. Turbid means it will look like cloudy. It is not transparent anymore. Normally, all the culture cells media looks transparent. But when it is infected with bacteria, the transparency will go and it will look cloudy. It will become yellowish. Normally the pH that we usually use for culturing any cell is maintained within 7 to 7.2. Then when bacteria infection will increase, then the pH will drop very fast. It will become like 4 or 5. It will become totally acidic. Bacterial culture makes the culture media totally acidic. So the color also, the media color, we usually use in a laboratory normally for pH, for recognition of particular pH, we use phenol red. All the culture media usually use phenol red as the marker of the pH. But phenol red is a little bit reddish to orangish in color. Then when bacteria infection spread, the bacteria density grows in the culture media, the pH drops. And when the pH drops, that phenol red become yellow. So the culture media, after huge bacterial growth, the culture media will turn turbid, cloudy, and the color of the culture media also will turn yellow. So these are the significant two markers to know that with naked eye we can recognize it. We see these two changes in any culture media, we can say that yes, this culture is totally contaminated with bacteria. And all this, if when we check the culture under microscope, if it is really cell debris, all those dots will not move. But when it is bacteria, spiral or rod-shaped or round-shaped bacteria is present, they will be motile, non-stop they are moving. The cells doesn't move, cells will stay in one place, but the bacteria, if it is contaminated with bacteria, the bacteria will be moving non-stop. Non-stop, it has non-stop motility. So from this, we can identify that the culture media is already contaminated with bacteria. So these are the story about the bacterial contamination. Next, bacterial contamination is the most common type of contamination in our cell culture technique. We almost every time we face this kind of contamination. Now, if we go for the next contaminant, that is another very common type in cell culture, it is mold. So mold is from fungi family. It is a multicellular organism, and it is filamentous, and it is much larger than bacteria. So normally when mold infect the culture, it usually, like bacteria, has very fast growth rate. Within, if we bacteria infection start within 2 to 3 days, it will the density of bacterial culture will become too much high, and it will be visible through naked eye. But mold is something that doesn't grow so fast. It will stay calm, quietly, and patiently in our culture media without letting us know that this unwanted uh, organism is already present in our culture. So if mold infection happen in our culture media, it will go inside the culture media and stay there for longer period of time, like 4, 5, even 1 week. It can stay in the culture media. Then after that, so we will be subculturing our cells, but we will not know that mold is there. Maybe under microscope we will see small small dots, but until the density of the mold become too much high, we cannot see it with naked eye, or even under microscope also it will not be too much visible because it doesn't has much motility also. As in bacterial initial infection, in mold initial infection also we will consider it as the maybe some cell debris, maybe some cells are broken, so the dots are coming. We will just consider it like that. But suddenly when it will start growing slowly, but it will continue to grow. Then what will happen? The media will become turbid, start turning in turbid. The cloudy appearance of the media, which usually happen in bacterial contamination also, that same turbidity will be coming for mold infection also. And in this case, in mold infection, the pH of the media increases. Like bacterial infection, the phenol red color turn into yellow, and because the bacterial infection lowers the pH. But in case of mold, the pH of the media increases, and as the pH of the media increases, the phenol red color of the normal natural color of the healthy media will turn into pink, totally pink color. The media will change into totally pink color, which is the alkaline type of color of phenol red. So from this, we will know that some kind of normally not only mold, any fungi infection, the pH increases and the color of the phenol red color changes into pink. Why the pH increases in fungi and why the pH decreases in bacteria? Because in bacterial contamination, the kind of toxins released from the bacteria are acidic, so which lowers the pH of the media of bacterial infection. But in case of mold, the toxins released are alkaline, so which increases the pH of the cell culture media and turned it into pink color. So when after few days, as the infection, as the mold will be growing inside the culture, as the infection start growing, then after few days, when we check the flask under microscope, we will see what these kind of patches are usually seen under under microscope. In case of mold infection, it will be like big hairy patches because it is bigger in size and it filamentous. That is why this kind of hairy patches we will see in case of mold infection. So this is a kind of mold infection, and all this kind of fungi infection normally happens mostly in monsoon time. The very source of fungi infection is normally from the water bath. I told you earlier that in incubator where we keep the cells, we have to keep the home of the cells moisture. We have to keep it moist. So for that, keeping that cells home moist, what we use is water bath. In one tray inside the incubator, we keep sterile water to keep the whole incubator moist. So from that water, generally, most of the time, this fungi infection usually happen. That water also get contaminated with fungi very easily, and fungi always like little bit watery place to grow. So if any fungal infection start in the water bird, then all the cells that are kept inside that incubator will get infected from the same fungi. So this is the story about the mold infection. So now we'll go next into another very common type of infection, which is viral contamination. Virus is, as we know, that it depends on, it cannot live on its own. It needs a host body. So it is a very small in size and very difficult to detect. When we detect virus in our cell culture, in our culture flask, already maybe all cells are gone, because if virus infections start in our cell culture, it will go and start infecting the hosts, all the cells in the culture media, and what will it do? It will grow and disrupt the total cells. So virus also initially we will see that small dots, maybe dots also we cannot see. Virus infection is very fast. If virus infection start today, maybe within 2 days, all cells are gone. And virus, I cannot say that it is very common like bacteria or mold infection, but it also happens. Especially if the biologist who is working as any kind of viral infection, like sneezing, coughing, or anything the laboratory worker or the biologist is doing, then from the nose or mouth of that biologist, viral infection can spread. But it is not as common as the bacterial and fungal infection in the cell culture. So it is very, so this is, virus cannot be detected under microscope also. Under microscope, the way we can see is fungi or bacteria like dots. We cannot see that kind of virus under microscope. When the whole cells are gone, only then we will see the culture media is full of dots like this. These are the normal cells growing like it's like a leaf structure. These are macrophages which are adhere with the culture flask. But when any viral infection happen, everything disrupted. All are destroyed. So just dots, cell debris, and all are only there. No intact cell will found. So this is all about virus. Now another fungi family, another very common infection in cell culture is yeast. It is also from fungi family. Its size is bigger, 2 micrometer to 40 micrometer, quite big, almost like the cells. Almost it is quite big and almost like the cells. So this yeast also has almost same characteristic like the what did we say, like the mold. It infect the cell and stay there quietly, gently, with patience for a few days. We'll be doing subculture. We will be subculturing our culture, and but still we will not know that already yeast infection has started. But suddenly, not suddenly, after for a long time, when yeast will continue growing, then maybe one fine morning you will notice that our culture media has turned into turbid, and also in like in case of mold, yeast also increases the pH of the culture media and turn the phenol red color into pink. So from the turbidity and the pink color, we will know that something wrong has happened. Maybe like any fungal infection has happened inside the cell culture. So when we will go to see it under the microscope, we will see this kind of spherical yeasts inside the cultured cells. So next is about mycoplasma. Mycoplasma is also a very common contamination, and it is what will I say? It is the maybe the most common type of contamination that usually happen nowadays. Mycoplasma is also basically a bacteria, but it is very small in size, the smallest among the total bacterial group, and it doesn't has any cell wall, and it grows very fast. And it's normally bacteria's diameter is 1 to 10 micrometer, but it is less than 1 micrometer in diameter. It can be detected only when the density becomes extremely high. Even mycoplasma, when mycoplasma infection happens, it also will look like dots. Initially, we will not know that mycoplasma infection has happened because it is so small. Even under microscope, we quickly will not become able to notice it. When the concentration increase a bit, maybe then when we will see it under microscope, see the culture under microscope, we will see that the few dots, we will notice that some dots are there, small dots are there, and but still cells will be growing. Normally other bacterial infection, in other bacterial infections, as the bacterial infection spread, the cell's growth retarded. The cell, the way it usually divides, the growth rate will be retarded. But in case of mycoplasma infection, cells continue to divide. Cells will continue to grow. It will almost like normal, normal growth rate, the way it usually grow without any contamination, almost in same way it will be growing in the initial infection time of the mycoplasma. So we will not know that already some infection has happened. But after some time, when the mycoplasma density will become higher, what will happen? Mycoplasma in growth, the number of mycoplasma will be increasing, and the number of cell will be decreasing. Number of cell will be decreasing means the cell growth rate will be retarded. The way cell usually grow, that growth rate will not be maintained anymore when the mycoplasma infection will be higher. So, as a mycoplasma infection continue, we will like maybe with mycoplasma infection we can culture the cell for 1 or 2 weeks even, but without knowing that something infection has happened, bacterial infection has happened. Without knowing that, maybe we are subculturing the media. But when the density, mycoplasma density becomes extremely high, then the total growth rate of the normal cells will reduce, and cell will be there, it will not be growing, it will not be dividing in their normal rate. From this, we will come to know that something wrong has happened. Then gradually we will see that the pH of the culture media will also be decreasing, as like all other bacterial infection. Mycoplasma also decreases the pH of the media, and the culture will turn yellowish, and cloudy appearance will also happen. But in other bacteria, the way the media turn turbid and cloudy appearance, all those things will not happen so fast in case of mycoplasma, because it takes time. It stays inside the culture for longer period of time than other bacterial contamination. So, apart from all these biological contaminant, there is another biological contaminant which we call cross contamination. What is cross contamination? It is like we are growing uh, particular cell, like we are growing leukemia cell line, and if suddenly inside leukemia cell line one or two lung cancer cell comes inside, and it also start growing with the leukemia cells, then this is known as cross contamination. Cross contamination means when we are maintaining one particular type of cells and any other type of cell line, not any other microorganism, any other type of cell line, just enter in that cell and reside there, and we start subculturing together the two types of cell lines, then it is known as cross contamination. Cross contamination usually happen by the mistakes of the biologist. When we are maintaining like 2-3 different types of cell lines together, and we are just subculturing the 3 4 2 3 different types of cell together, then by mistake maybe uh, our one cell can be contaminated, can enter into, by mistake we can put one type of cell in not their specific class, in another class we can put, and two cells just mixed. So this is how the cross contamination can happen, and it is a very common problem nowadays, because in one laboratory we usually maintain so many types of cells for our different types of experiments. And cross contamination, it is said that nowadays maybe like 15 to 20% of the cells that biologists use all over the world are cross contaminated, which is also not expected, because if we want to work on lung cancer cell line and any cervical cancer cell line cells also enter in that lung cancer cells, then what the expected result that we supposed to get from the lung cancer cells, we will not get that, as the cervical cancer cells are mixed already with the lung cancer cells. So this is also a major problem. So we have to be very cautious when giving subculture of the cell lines, so that all this kind of mistake of cross contamination should not happen. So for this, maybe I'd like to maybe go for break now, Sungita.
Yes, you can we take a break for 10 minutes?
Yeah.
Okay, students, now the break time. We will meet after 10 minutes, the same platform. Okay. Am I audible? Hello, am I audible?
Ma'am. Yes, ma'am.
Okay, Dr. Datto, are you available?
Yes.
Okay, so welcome back, Dr. Datto. Please continue with your presentation. And students, if there is any query, please write down in the chat box. We'll place your question to Dr. Datto after the session. Okay, Dr. Datto, please continue with your presentation. Thank you.
Okay. So thank you, Shungita. So welcome back, all of you. So far, what we learn about the cell culture and the contamination, biological contamination, let us just give a brief recapitulation of that. So what we know about the contamination is presence of any type of impurity or unwanted element in our culture media is known as contamination. And there are two types of contamination, biological and chemical, but we mostly bother about the biological contamination. And biological contaminants are mainly of five types: bacteria, virus, mold, yeast, and mycoplasma. So among these, bacteria are in the same group, and mold and yeast are from the fungi family, and virus is separate. So what we learned that common causes of contaminations can be from culture room air, or from the hands and clothes of the biologist, or from the media and FCS that we are using, or from the plastic wares that we are using inside the laminar hood. So among different type of contaminant that we spoke about, five types of contaminants, the most common ones are bacteria, and bacterial contamination, other bacteria, and mycoplasma also. Mycoplasma, why we are putting mycoplasma, though it is in bacteria group, but we are talking about mycoplasma separately. Why is it so? Because mycoplasma is a kind of bacteria that doesn't have cell wall, and it is much smaller in size than regular bacteria. So we know that bacteria and mycoplasma, both contamination reduces the pH of the culture media and turn it into yellowish and make the culture uh, turbid. And both of them grow very fast. And yeast and mold, these two types of fungi, these two are from fungi family. They usually increases the pH of the culture media and make the culture turbid, and their growth rate is usually slower than the bacterial growth rate. And virus infection usually uh, it is not so common, but we face viral infection also in our culture media. And this viral contamination, when it happens, it just destroy the whole cell, because virus cannot survive independently, so it need a host body. So it uses the cells in the culture media as their host and disrupt the host body, and they start growing enormously. So then we talked about cross contamination also. That is another type of biological contamination in which two different types of cell lines mix together, and we continue growing them together. So the expected result that we want from the particular cell line, we may not get that result, as it is already cross contaminated with another type of cells. So now what we are going to do, we are going to discuss about the most important part of this, that is precautionary measures that we have to take to prevent all these contamination issues and do our experiment successfully. Bacteria, yeast, virus, their growth is not in our hand, but from our mistakes, our unintentional mistakes, these contaminations happen. But we are wiser than all these microorganisms, so we have to use different precautionary measures so that all these contamination should not happen and destroy our experiments, destroy our hard works. So for this purpose, what we used to do, first of all, the biologist who is doing the subculturing or the cell culture work inside the culture room must wear gloves in our hand, and the gloves that we wear for the cell culture should be use and throw. It is not that we will be using the gloves day after day. We will use it once, today we will use it and discard it. Then next day we will use another gloves. And lab coats are also very important. And lab coats should not be taken back home or kept with other things outside the culture room. Lab coats should be kept when we are entering the culture room. We should put on our lab coats, and when we are we are going to exit from the culture room, we'll just remove our lab coat and keep it inside the culture room in one place, one shelf. And after even wearing the gloves, we have to use 70% alcohol to sterilize our hands. Though we put on gloves, but still we need to use alcohol to sterilize our hands. And it is mandatory that the hood, laminar hood, should be used whenever we are doing any subculturing or we are opening any cultural flask or anything. So laminar hood is, as I told initially, that this is the mandatory instrument that we must have inside our culture room. Why laminar hood? What is the function of it? We know that air is there inside the culture room, which might be contaminated with the biologist who is working there with the cough or sneezing or the talking of the biologist. So this airborne contamination should not be transferred to our culture media. That is why, as a precautionary measure, we use laminar hood. What is the function of laminar hood? Laminar hood take the air from the culture room and purify it, sterilize it, then blow it towards outside. And as it is blowing outside, it is not allowing the outside air to go inside. So in this way, purified air is always blowing inside our working bench in the laminar hood. So in this way, the airborne contamination, we can reduce the chance of any kind of airborne contamination by using this laminar hood. And it is compulsory that all sort of culturing, subculturing or cell culturing work should be done inside the laminar hood. But when we are using the laminar hood, we have to keep in mind that this is the most important instrument that we are using, and it is a preliminary precautions that we are using while we are opening all our cultural plates, flasks inside this hood. So this hood should be maintained properly. It should be cleaned, or it should be maintained with the proper technicians every time. It is like a filter. So all those filters need to be cleaned regularly, maybe after 2-3 months or 4 months. After 4 months, regularly with experts, the filter should be cleaned properly, so that no airborne contamination we should face any kind of airborne contamination. Now the next one is incubator. Inside incubator, we should always regularly, maybe like after 2 to 3 days, we should clean the incubator with alcohol. And as I told before, in incubator, we have to keep the home of the cells very moist. Moisture condition should be maintained. So to maintain that moisture condition, what we usually do, we have a tray. We usually have a tray, metal tray inside the incubator, inside which we keep autoclaved water, distilled water, autoclaved water. We keep inside the incubator, which is known as water bath. But this water should be changed regularly, because as this water is kept in open tray, and water is very prone to fungal infection. Fungus love to grow inside water. So this water bath should not be kept for longer time, and should always be whenever we are cleaning the water bath and putting new water in it, we should add some antifungal agent like gentamicin, like gentamicin also we can add, or like one detergent SDS also we can add, so that no fungal contamination should happen inside the water bar. And also we must change this water regularly after a certain period of time. Maybe once in a week we should throw away the water, clean the plate very well, and put fresh autoclaved water and add fresh SDS or gentamicin to it, so that fungal growth can be avoided. Now, another thing which is mandatory is 70% ethanol, or 70% ethanol is most preferable inside in the cell culture laboratory. Now, why 70% alcohol? Why not 100%? Say 100% should be better than 70%, but no, 100% is not better in time of sterilization. Even nowadays, as we are using sanitizer, we are habituated it has become part and parcel of us to use sanitizer, hand sanitizer, and sanitization of surface and sanitization of every outside things. In that case also, it is 70% alcohol. It is not 100%. But why this 70% alcohol? Because in time of sterilization, 70% is far better than 100%. How? If 70% ethanol is used, that means 30% is water, and this 70% ethanol, as it is with 30% of water, so it is surface time. Surface time means when we clean the laminar hood with this 70% ethanol, the time the ethanol surface time will be this 70% ethanol will be on the surface of the laminar hood is much more than the 100% ethanol. If we clean the surface with the 100% ethanol, instantly it will evaporate. But if we use 70% ethanol, it will take small time before it will be completely evaporated. So surface time is higher. And also not only this, another thing is 70% ethanol, when it comes to in contact with any kind of microorganism, like our enemies, all these contaminants, what happens? It enter slowly inside from the surface of the microorganism inside and coagulate the proteins and microorganism proteins, and this entering into the microorganism body is in slow rate and coagulate the protein slowly and destroy the microorganism completely. But in case of 100%, if the alcohol is 100%, this interaction of that 100% alcohol and the protein of surface protein of the microorganisms is very fast, and it might not kill fully. The microorganism may not be killed fully. So 70% ethanol slowly absorbed to the microorganism surface and enter the microorganism body and coagulates the protein and kill the microorganism very successfully. But in 100% ethanol, the protein, microorganism protein and the alcohol interaction will be very fast, and it might not kill the microorganism completely. So for better sanitization, better sterilization, we have to use 70% ethanol, not 100%. It has to be kept in mind that ethanol should be used 70%. We have to mix the bottle that we buy from outside. It is always like 99%. So we have to make it 70% before using inside cell culture laboratory. And this ethanol is compulsory in the laboratory, in the cell culture laboratory. Anything we want to use inside laminar hood, whether we want to put our hand inside the laminar hood, we have to sterilize everything with 70% ethanol. Even the laminar hood, the desk where we used to do the cultural work, we have to clean it with 70% ethanol always before starting our cell culture work. And also when we want to clean the incubator, we have to use 70% ethanol to clean all the doors and shelves, everything of the incubator. We have to use 70% ethanol. So now next is sterility. Sterility of the things that we are using inside the laminar hood for our cell culture purpose. What we are using? Mostly we are using plastic wares. Plastic wares means micropipette, then tips, then tubes, then plates, petri plates, flask, append or small micro tubes, like this. But everything should be sterile. How? Sometimes nowadays we usually all the tips, flasks, tubes, whatever we use in cell culture purpose, we use it once and throw it away and discard it. But before also, when it was not so, if now, even now also, if the funding of the lab is not so much high, then what we do, we reuse the tips, flask, tubes, petri plates, everything. How to do this? We use it, then wash it with acid. We either we make it 1% ethanol, HCl, hydrochloric acid, or we make formic acid solution in laboratory. We wash it with acid, then we autoclave it. So all this plasticware need to be, if we are using this plasticware, we need to clean it properly, then autoclave it. And if we are using use and throw technique, then all the plasticwares nowadays packed in sterile way. So we open the packet inside the cell culture hood, we use the plasticwares and throw it away, discard it after using once. So there is no need to autoclave or washing or anything. So these are the two methods by which the plasticwares should be sterilized. Another thing is the things that we use inside culture. For culturing purpose, we use culture, as I told before, that we use culture media and fetal calf serum. So FCS, that is FCS. FCS is for the nutrition of the cells, and media is for the growth of the cells. So these two things also, we usually, we purchase it ready-made from the company, liquid media and FCS. And for every cell culture, we usually need to add 10% of FCS to the culture media. So this 10% FCS and culture media, this FCS and culture media also should be sterilized, but we cannot autoclave them to sterilize. It will totally destroy everything, all the components will be destroyed. So what do? We do filtration. We use 0.22 micron filter paper to filter the media and the FCS. 0.22 micron means it's supposed to prevent all the bacteria, virus, fungi to enter into the purified media or FCS. So everything will be removed by using 0.22 micrometer filter paper. So plasticwares are clean, the media and FCS are also clean. Now what? Now the next thing is how to purify the air of the culture room. For purifying the air of the culture room, what we used to fumigate. We used to fumigate the culture room. And what is fumigation? The culture room, as I told, that it is a completely separate room. So this particular room has to be fumigated. And how to do this? We used to use formaldehyde for purification. We used to use potassium permanganate. We keep it inside one bowl and add some formaldehyde into it and close the door. And close the door completely of the cell culture room. So immediately as you add the formaldehyde to the potassium permanganate, the fume will start coming out, and the whole room will be full with fume. that fume. This fume is not good for our health. This fume is very toxic, and it is very bad, very unhealthy for our lungs. So immediately we have to close our nose with mask and just add the formaldehyde and come out quickly and close the door of the culture room, so that the fume should not enter through our nose. We have to keep the culture room like this overnight, and next day, so the fume will fill the room and go away from the room. Then next day we should remove the bowl inside which we give for fumigation. This fume is highly alkaline and which kills the microorganism. If any microorganism is present inside the culture room, this fume kills the microorganism. So this fumigation process, through this fumigation process, the air of the culture room is purified, and this fumigation process is very effective, and we have to do this thing regularly, like once in a week. We must do this inside our culture room, so that no airborne contamination should happen. And another thing is, during the monsoon time, all this cell culture contamination rate increases. It is always very, there is all the cells, culture media, everything is prone to contamination during the monsoon mostly. So during monsoon time, we must do this fumigation regularly and change the water bath of the incubator also regularly, maybe more often than all other time of the year. So this is the procedure of fumigation. Now, next, another precautionary measure that we used to take for the cell culture is using of antibiotic. For antibiotic, the most common antibiotic for most of the bacteria is penicillin and streptomycin. So the common bacterial infections that we usually have, we can as a precautionary measure, we can add penicillin and streptomycin mixture in our culture media when we are before putting the culture media in to the cell. So this antibiotic should not be used in should be used in a limited amount, in a permissible amount. Why? Because if we use higher concentration of antibiotics, it might do what? It might grow resistance or some kind of mutation among the cells. So antibiotic, whenever we are using antibiotics inside our culture media, the amount should not be more than 1%. So penicillin and streptomycin mixture is a common antibiotic solution that we used to use in our cell culture technique, and this is regularly used, and the amount is always 1% or less than 1%, never more than 1%, so that no mutation should happen to our cell line. And apart from this penicillin streptomycin, there is another antibiotic that we usually use, that is basically antifungal agent, which is known as gentamycin. Gentamycin, they sell it in like ampoules in regularly. It is usually available in all medicine shops. Gentamycin, it is like a capsule. One capsule we have to use for 1 liter of media. So gentamycin is for all type of fungal contamination. So gentamycin also we can add for the as a precautionary measure to our cell culture media. But antibiotic, but it has to be kept in mind that if we add antibiotic, streptomycin, penicillin, streptomycin or gentamycin, that doesn't mean that culture media cannot have any kind of contamination again. Because many bacteria nowadays, they are very clever, they are always mutating. And many bacteria, fungus, because they know that usually we add this kind of antibiotics and antifungal agent to our culture media. So they do what? They just mutate themselves and they start growing inside the media where already antibiotic or antifungal agents are present. So if we add all these antibiotics or antifungal agents, still we have to be extremely careful and take all other necessary precautions that we already mentioned. We have to maintain all these necessary precautionary measures we have to maintain so that we can save our culture from the from this from any sort of contamination. So now in brief, what are the precautionary measures I told? Firstly, we have to wear. Another precautionary measure that I forgot to tell is UV radiation. What is UV radiation? UV radiation, I think all of you know that already. In physics or chemistry, we have already learned that UV radiation do mutation. And how does it do mutation? It makes thymine dimers and stop the replication. And this is how it kills the microorganism. And in cell culture laboratory, we usually have a UV tube. It looks like the normal tube light that we used to use in our long tube light in our home. That same kind of long tube, but it is not white in color. It is transparent. And this UV radiation, we have to any time if we want to do subculturing of the this UV radiation, this UV lamp not only we put in our cell culture room, but also inside laminar hood, the hood desk where we are going to do our main cultural work. In above that hood desk, there is also an UV lamp. So before using the laminar hood and the culture room, we must switch on the UV lamp, both inside the laminar hood and of the UV lamp of our culture room. We should leave the UV lamp on for like 30 minutes so that it is another precautionary measure. So before subculturing, for 30 minutes, UV exposure of the room and laminar hood and is also very important as a precautionary measure. So this is another extra, like UV lamp is used for, as I told, the fumigation we use for killing the airborne microorganisms. So this UV lamp also we use for killing the all the microorganisms present inside the air of the culture room or inside the laminar hood also will be killed by this UV radiation. So as a sum up, what are the precautionary measures that I told now? The biologist must wear gloves, use lab coats. Laminar hood should be used for any kind of cell culture work. And the laminar hood not only used, but it should be maintained properly, because this is the most crucial equipment under which in the desk of which we are opening all our culture, we are opening the flask of our culture, the totally culture, all the cell culture is exposed in this area. So laminar hood should be maintained properly. And the incubator also should be cleaned regularly with 70% ethanol. And the water bath inside the incubator should always be cleaned properly because that water has a high chance of fungal infection, fungal contamination. So we should maintain the water bath very particularly, very cautiously, and change the water of the water bath regularly. Then fumigation is another step. We should fumigate the culture room regularly. Then antibiotic and gentamycin should be used as a precautionary measure to the cell culture media, but in a permissible amount. And anything we are putting inside the laminar hood should be sterilized with 70% ethanol. So these are the basic precautionary measures and very crucial to keep in mind before we are entering into the cell culture system. Now, these are the precautionary, but if whether there is any chance, if any culture has got bacterial or fungal infection, is there any chance that we can uh, overcome the contamination, we can destroy the contamination and take out our desired cells? There are some methods that we can use, like if any culture we know that bacterial contamination is there, we have checked it under microscope and we have confirmed that it is bacterial contamination. What we can do, but and we know that that culture is very important for our experimental work. We can try. What we can try? We can add some antibiotic like streptomycin and penicillin mixture to it and keep it for like 1 or 2 weeks. Not keep it totally, but we have to be doing subculture in it. Like after every cell has their different growth time. Some cells grow in 24 hours, some 48 hours. So accordingly that we have to do subculturing of the cell lines. Some for after 24 hours, some after 48 hours, some after 72 hours. Accordingly that after always we have to do subculture and continue adding the antibiotic to the culture, to the contaminated culture. So after like 1 or 2 weeks doing subculturing like this, we have to check if the bacterial contamination is reduced. Bacterial contamination, bacteria are not growing more and the cells are growing, gradually start growing. If this thing happen, then we will know that maybe the contamination, we are becoming able to overcome the contamination. So we have to pellet down, we have to spin the cell culture and pellet down the bacterial contamination and separate it and take the cell, our desired cell, from the top of the tube. Then if fungal contamination is there, then we can do what? We can add some, same way we can add some gentamycin, extra gentamycin and culture it, continue culturing it and adding the gentamycin every time we are doing subculture, and we can monitor the flask for like 1 to 2 weeks and check if we are becoming successful to overcome the contamination. But in case of viral infection, no, there is no chance of overcome, because virus, if any virus infection happen in our culture media, virus will destroy the whole cell. All the cells will be burst and there will be only virus and cell debris inside the culture medium. So in case of viral infection, we cannot try even all these kind of techniques. But still, what is preferred, that if any kind of contamination happen, then the flask, the petri dish should be discarded. We should autoclave it and throw it away. With maintaining proper protocol, we should throw it away. By adding this antibiotic, by adding this antifungal agent, maybe it will we can get some fresh, good cells without contamination, but there is high chance that contamination will start in that, and we don't know if the cell genetic morphology will be remain same after the contamination. So it is better if we have option, if we have our backup of those cells, then we should just throw away the contaminated culture, autoclave it. Throw away means we have to maintain proper protocol. We have to autoclave the culture, destroy the contaminant totally, then dispose it. So another important thing as a precautionary measure that we can do is freezing of the cells. We can freeze the cells, the cell line, maybe if I want to work with HeLa cells. HeLa cell, as I showed in my slide, that it is a cervical cancer cell line. So I brought it from the cell bank. So after bringing it, I will culture it. So when I will see that fresh cells have grown and lot of good healthy cells are there, what will I do? Some of the cell I will take and freeze it. Freeze it means For freezing the cells, we use liquid nitrogen. It is very it is a place where we can save cells for like 6 months to 1 year, freeze cells for 6 months to 1 year, and the freeze cell after 6 months to 1 year, when we thaw it, when we put it in culture again, taking it out from the liquid nitrogen and try to culture it, maybe it will grow. Usually it will grow and become healthy again. So as a precautionary measure, what we can do, there is always chance of contamination. We all know that. So as a precautionary measure, we should, anytime we are using any cell, as a backup, so that if contamination happen, we will not be lost. We will not just be sitting down without doing the experiment. We should freeze the cells, some vials, like 5-6 vials, we should freeze and keep inside liquid nitrogen, so that if any contamination happen, no problem, just throw away the cell and take out another one vial from the liquid nitrogen, thaw it, and start using it like normal and do our desired experiment. So like this, we can overcome all these burdens of contamination. So this is all that I have to tell about the biological contamination, cell culture and biological contamination, which is a nightmare for all the biologists. I also have faced lot of this kind of issues, and it wasted lot of time during my PhD life also. I just hope that the students who are listening to it, you will keep it in mind, so that in future, when you will be a researcher, when you are going to do biological research, all these contamination issues should not bother you, and you will become very successful to avoid this contamination and do your experiment very successfully and be a very successful researcher. So that is all that I have to tell about the contamination. So now,
Thank you so much, Sanchita Di, for accepting my invitation. And your talk is very informative. We got a vast knowledge on cell contamination and learn how we can save our culture from contamination. Now the session is open for question and answer. Students, you may ask question to Dr. Dotto directly, or you can write down in the Gmeet chat box. Sanchita Di, one question from Shambhavi. You can read it from the chat box, or I can convey to you.
Yeah, please tell.
Yeah, the question is, then ma'am, how can we prevent the growth or the development of these molds, fungi?
It's not audible.
In the yeah, Dr. Arnabda, Dr. Dotto is there. Yes.
Okay. See, for any kind of contamination, the normal protocol is same. We have to use laminar hood, we have to use 70% ethanol, we have to use all these things. But particularly for fungal infection, what we have to do as a precautionary measure, as I told, that we can add some permissible amount of gentamicin in our culture media, and the incubator water bath. The water bath inside the incubator should be changed regularly, and should be fresh autoclaved water should be added in the water bath. And for prevention of the fungal contamination, we must add detergent like SDS, we can add. So and the normal procedure is same for fungal, for bacterial, for every infection it is same. And fumigation is important for both, as well bacterial and fungal. to avoid both the contamination. So procedure is same for all the contamination, but for fungal contamination particularly, what we do gentamicin. We add gentamicin in our culture media.
Okay, Shambhavi, I think you got your answer. Any other query from students? Any other query? Any other query, please write down in the chat box or ask directly to Dr. Dotto. I hope no more queries there. Thank you again. Thank you again for accepting my invitation. I hope to hear you again. And I hope that students enjoy a lot. So the next session will be on 8th in the same platform. The time will be from 5 p.m. as already mentioned in the brochure. So that is all for today. Thank you so much for all of your cooperation.
Thank you, Shongita, and thank you all the teachers of the college, and thank you, principal also. Thanks a lot for everyone for giving me this kind of platform. It is a pleasure for me to be invited in this platform. Thank you all. I will be looking forward for this kind of opportunity again.
Yeah, sure. Thank you.
Thank you very much.
Thank you. So, Arnabda, can we end the meeting? Okay. Thank you.